The cofactors in the enzymes. Now we had seen that these particular enzymes, rather most of the enzymes, they are protein, right? You remember that there are certain RNA molecules which act as enzyme. Yes, we call them as ribozymes, right? We'll be discussing about them much more in detail in some other video, right? Having a great importance in evolution and help us understand the origins of life. The hypothesis of RNA world, right? That is about those ribozymes. So remember, ribozymes are RNA molecules which are acting as the enzymes. Today we'll be talking about these cofactors in the enzymes, right? The protein part of the enzyme is the major part of the enzyme. We had seen that the tertiary structure of this particular enzyme that was responsible for catalyzing the reaction, it had that active side. But yes, in this major what we call as enzyme, which is a protein part, which we call as the apoenzyme, there is a small non-protein part which is called as the cofactors. So remember, the non-protein component of this particular enzyme is a cofactor. The protein component is the apoenzyme. So apoenzyme is the protein part, cofactor a non-protein part, but essential for its activity. You remove a cofactor from the enzyme, it is of no use if at all it is having a cofactor. So that testifies that these cofactors are really important for the functioning of these enzymes. Okay, so if this cofactor is very tightly bound to the surface of the enzyme, it is like covalent bound, you are trying to remove it, but you cannot remove it without destroying the enzyme. So if it is an integral part of the active site, it is very tightly bound to the surface of the enzyme, we call it as the prosthetic group, an organic molecule, right? So this prosthetic group is tightly bound to the surface of the enzyme. To take an example, you have the heme group, right, which is very tightly bound, integral part of the active site of the enzymes like catalase and peroxidase, which are catalyzing the hydrolysis of hydrogen peroxide to form H2O and water. Oh, H2 and water is same to form H2O and oxygen, right? So breaking your H2O, H2O2, right, that is your hydrogen peroxide, to H2O and oxygen, your catalase and peroxidase, they are having this heme group as the prosthetic group. The coenzyme, again a cofactor, which is again an organic molecule, but then this is transiently attached. It comes and binds to the enzyme whenever the reaction is to be catalyzed, right? So this transiently bound cofactor, that is your coenzyme, mostly again it is also a vitamin. You have this niacin. Earlier, this niacin was referred to as nicotinic acid, which looked like, or which sounded like nicotine, which is present in tobacco. So probably some people thought that tobacco is nutritious or what, so they're taking a source of vitamin, which is not true because that is nicotine, which is an component there, and here we are talking about nicotinic acid. Let us not call it nicotinic acid, let us call it niacin, a part of NAD, nicotinamide adenine dinucleotide, and NADP, the nicotinamide adenine dinucleotide phosphate, which acts as the coenzyme for various dehydrogenases, oxidoreductases, which we came across during Krebs cycle or glycolysis, which are acting as the transient carriers of the hydrogens. So it is only transiently bound. So whenever you want to do the reaction, that time it will be bound to the surface of the enzyme. Then it can leave the surface of the enzyme, go to your ETS, and so on and so forth. Anyways, some enzymes, they require metal ions, like to form a coordination bond. On one side they'll what you call as bind to the active site of the enzyme, on other side they'll go and bind to the substrate. They'll try to bring them together. It's like matrimony. So it's like bringing these two together, the substrate and your enzyme together by forming coordination bonds there. Many other roles they may play, like they may act as the actual inherent electron carriers, maybe. But then you'll find that one of the examples here is your zinc, which is a cofactor for what you call as the carboxypeptidase, which is a proteolytic enzyme, right, which is degrading proteins, right? So these are your cofactors. So to give a brief summary, the cofactors are the non-protein constituents. If they are tightly bound to the surface of the enzyme, it is prosthetic group. Heme group in catalase and peroxidase, which is catalyzing the hydrolysis of hydrogen peroxide to form water and oxygen. The coenzyme, which is transiently bound, an organic molecule, mostly vitamin. Example is niacin. In the form of NAD and NADP in dehydrogenases. Then we have metal ions forming coordination bonds. Example is your zinc in carboxypeptidase. So stay tuned with me with Girish Kukreja for more in biology. Thank you.